Hi everyone, my name is Hossein Rajabzadeh, and I've been working on the development of biocompatible hyaluronic acid hydrogels for nerve Nano-Clip fabrication. I had the pleasure of working with Jacob Townsend, Morgan Brown, Annie Gilbert, Professor Gardner, and Professor Hetirachi, who are affiliated with the Knight campus for accelerating scientific impact and the Department of Chemistry. The project was inspired by the Gardner Lab's use of photosensitive, semi-organic hormocomb and IP-dip materials to 3D print nanoscale implantable electronic devices called nerve nanoclips. These nerve clips were used to electrically stimulate the tracheopharyngeal nerve of zebra finches and induce audible vocalization. Beyond the study's success, the material composition of the nerve nanoclips also stimulated mild inflammatory response in the surrounding nerve tissue. For this reason, there is a need for a biocompatible material composition of the nerve nanoclips, which can be photo cross-linked on a nanoscale. Our objective was to develop a biocompatible minimal swelling hydrogel from hyaluronic acid, or HA, and that could be photo cross-linked for the fabrication of nerve nanoclips, and that would also cause a negligible inflammation and nerve tissue damage. To do this, we synthesized methacrylated hyaluronic acid using an esterification reaction with different molar excesses of methacrylic anhydride to HA monomers under basic conditions. The degree of methacrylation was quantified with Proton Nmr Spectroscopy using the Broker AV500 by comparing the integral peaks at 2.05, 5.78, and 6.21 ppm, which pertains to the HA backbone methyl protons and methacrylate alkene protons, respectively. A solution composed of 10% photo initiator, Hydrogel 2959, and a 4% methacrylate hyaluronic acid was prepared in phosphate buffered safety and exposed to ultraviolet light for 5 minutes to form photo cross-linked hydrogels utilizing radical chemistry reactions. Methacrylated hyaluronic acid conditions were cut into cylinder and square geometry samples with a volume of 50 microns. Each sample was weighed at initial post-gelation time of 0 hours and increasing time points of 4 hours, 6 hours, 1 day, and through a 10-day span. The absorption ratio was calculated by dividing the weight at different time points by the weight at 0 hours. The resulting absorption ratios were then averaged and compared to an ideal absorption ratio of 1, which indicated minimal swelling. Our results indicated that the successful synthesis of MIHA was confirmed by Proton Nmr Spectroscopy due to the appearance of peaks at 5.8 and 6.21 ppm, which pertain to the methacrylate alkene protons shown with green dots in the spectrum of 2.5 times, 5 times, and 10 times conditions, and the absence of these peaks in the hyaluronic acid spectrum. The transition in peak intensity from 2.05 ppm shown in red to 1.98 ppm shown in blue, which pertains to the methyl protons on the HA backbone and the methyl methacrylate protons, respectively, also confirmed successful MIHA synthesis. Proton Nmr spectroscopy revealed that the levels of modification of 2.5 times and 5 times and 10 times MIHA were to be 30%, 46%, and 54% respectively. MIHA hydrogels of 50 microliters were successfully photo cross-linked in the presence of Hydrogel 2959 photo initiator under UV light. MIHA hydrogels with a volume of 3 microliters were also photo cross-linked with the Gardner Lab's 2-photon 3D printer. The average swelling ratio of 2.5 times, 5 times, and 10 times MIHA hydrogel conditions over a 10-day span that were calculated to be 1.01, 1.01, and 0.92 respectively. This data suggested that all MIHA conditions swelled under 10%, indicating minimal swelling. From this study, we can conclude The hyaluronic acid biopolymers can be functionalized with methacrylate groups using an esterification reaction under basic conditions to form MIHA. All MEHA conditions were highly photosensitive and could be cross-linked in the presence of photoinitiators under UV light, indicating that MIHA hydrogel solutions is a promising candidate for 3D printing complex geometries and on a nanoscale. The minimal swelling of MIHA conditions suggests that MIHA could be amendable for use in nerve nanoclips and will support reduced nerve tissue damage. Future studies could determine the neural stem cell viability and level of conductivity with MIHA medium before doing in vivo trials. Here are some of my references, and I'd like to acknowledge that this study was funded by the Hirachi Lab, Rural Community Grant, and College Medical Trust. Big thanks to CAMCOR staff for providing me with technical support and skills to complete this project. I'd like to thank the Office of the Vice President for research innovations and all work resulting from this research.